everybody, and this is Unit 5, Heredity. This is one of my favorite subjects, mostly because it's something I have studied and worked with for quite a bit. I'm excited to share it with you guys. You might notice as we go over some of the stuff, some of it might be a review, and that's okay. It's good to kind of remember these things and get it back into your head. Some of it might be totally new, so hopefully there is some new stuff for you in this unit. Okay, so I want to talk a little bit about some terminology. In particular, I want to just kind of clarify words like haploid versus diploid. All right, and what I already noticed in the slide, and I'm going to correct it for you guys, I have it written here as haploid. This is actually supposed to be diploid. And I have here diploid, and this definition actually right here is supposed to be haploid. So good thing I checked that out right away. But when I talk about the words haploid and diploid, the first thing I kind of want to talk about is what ploid is representing. Ploid represents the sets of chromosomes. And basically, you can have one set of chromosomes or two sets of chromosomes. There are some plants that can have 4 sets of chromosomes or 8 sets of chromosomes. And when we deal with that, we're talking about the ploidiness of that organism. So if you're a diploid, you are either an organism or a cell that has 2 sets of chromosomes, and we usually symbolize it with a 2N. The N represents the set. 2 means 2. And again, this is how we represent the diploid organisms. Some organisms can be, like I said, plants can be 4N, which is a tetraploid. In some cells, in the humans, we do have some sets of cells that do have only one set of chromosomes. Those are haploid, and we usually represent those with N. So that's just kind of some brief terminology that you might see throughout these conversations. And like I said, there are some cells in humans that have one set of chromosomes. Those are our gametes. So again, most of the cells in the human body are diploid, but the gametes are your cells that have one set of cells. And again, that's just a fancy scientific word for your sex cells. In humans, the sex cells are the oocytes or the spermatocytes. Those are the ones that make the eggs or the sperm. So why do we have those haploid cells? Well, really that comes down to the fact that when you are making a new organism, your parents are going to contribute a gamete, sperm or egg, with one set of chromosomes that will be paired with a set from the other parent, and that would hopefully get that new diploid cell or zygote formed. So now we've gone over some of the basic terminology. Let's go back to Gregor Mendel, who many of you guys know is the father of genetics. There's a lovely picture that many people have seen already before of Gregor Mendel. He's been giving peas a chance since 1866. So what did he look at? Gregor Mendel was really looking at traits in the organisms. He did not know about genes. He did not know about locusts. I don't know if he knew much about alleles, but he knew about traits, and he really studied those traits and explored them and did some crosses with them. And again, just kind of some terminology. If you don't know what a trait is, it's the express characteristics of your genes. Genes are chunks of DNA that could code for a particular recipe. Locus, or in plural, you might hear it as a loci, is the position of the gene on the chromosome, and then alleles are the alternate versions of that same gene. All right, so, things that we also know is that diploid organisms usually have 2 copies of each gene, one for each homologous chromosome. And if you're not sure what a homologous chromosome, well, we have some pictures of homologous chromosomes here. Like I said, you get one of the homologous chromosomes from your mother, and one from your father, or one from the egg, one from the sperm. And basically, they are chromosomes that have the same size and shape and contain the same genes, but they might have different versions of those genes. So again, you might get a trait from your mom, the maternal side of the family. You might get a trait from the paternal side of the family, but you're going to have the same type of gene on those chromosomes. And these are just examples of some of those chromosomes. This karyotype does have an abnormality. Can you find it? Now, when Mendel did his experimentation, he did use peas as his model organism, mostly because one of the things, and I'll talk about it later, is that geneticists love to mate things and see what the results are. And it's really hard to do that with lots of animals, especially humans. So we try to use things that we can manipulate without getting a lot of people complaining, and also gives us lots of offspring. So he looked at lots of different traits, and he noticed that when he mated his plants, that some of the traits were observed. He was looking at the observations. Those are what we call now the phenotypes. So basically it's what the organism looks like. He did not know about genes. He did not know about DNA. He did his work in 1866. DNA, as our genetic material, did not come out as a thing until 1950s. So about 100 years ahead, not exactly, but approximately 100 years ahead of the discovery of DNA as a genetic material. But now we know about genes, and so we have the genotype basically is the genetic makeup of the organisms. And again, just to help reiterate some information, homozygous are when you have two identical alleles for a trait, and heterozygous are two different alleles for a given trait. Like I said, geneticists love to do crosses. So they do these crosses by mating hypothetical organisms with specific phenotypes and genotypes, and they can do this on paper, just see what they get for results, and then they usually try it out in the lab to see if it works. Okay, so how do we do these crosses? It's important. As you want to label each generation of the cross. The first generation is the P generation for parental. Offsprings are usually F1, F2, etc. for filial. It's always good to make sure you write down the symbols that you're using for alleles with capital letters for your dominant traits and lowercase letters for your lowercase. So that gets us to start talking about what is Mendelian genetics. These are the genetics that followed Mendel's experimental rules and are pretty much the simple form of the genetics. Again, for many of you guys, this is going to be a review. For some of you guys, it may not be, or you forgot it. So again, this is something that if you want to fast forward, you can. But if you don't know it or you want to review it, it's a good thing to kind of review. All right, so one of the things that Mendel came up with was what he called the law of dominance. Basically, he took his plants, all right? Here we have a violet flower plant with a white flower plant, and he mated them because, again, geneticists like to do crosses. When he mated them, he saw that all the first generation plant were purple. He's like, okay, cool. So purple is going to be dominant to white because white disappeared, purple was there. One thing that he did a little bit more intriguing was that he selfed the plant, meaning he took the pollen from the plant and used it to fertilize the egg, and that was self-fertilization, and he ended up getting, in the next generation, lots of violet with some white flowers. So again, about 705 violets, 224 white flowers. And this kind of said that, yes, there's a trait that is dominant over the other trait, that masks the other trait, and that unmasking can be revealed in the F2 generation. Now, what Mendel did in this type of cross is what we call, actually this cross right here is what we call a monohybrid cross. So again, some definitions and some terminology for you. Mono, for as many of you guys know, means one gene. Hybrid is basically different variations of the gene. So remember I talked about alleles? This would be a monohybrid organism. It has a capital letter, so the dominant, and the recessive. This is a good terminology to keep in mind because sometimes you might get a genetics problem that will say, you take a monohybrid cross and what is the expected progeny, and you are going, wait, I don't know if it's homozygous dominant, homozygous recessive, or if it's heterozygote. Just using the word hybrid should tell you that it's heterozygote. Okay? So that's something to be really aware of. Now, usually when we talk about genetics, we always talk about Mendel, and you guys, nobody looks like you've seen a picture. But here's another person who was really, really instrumental in genetics. He came after Mendel, and really what he came up with was a way to represent some of the stuff that Mendel did. This is Mr. Punnett. In fact, this is Reginald Punnett. It's kind of fun to just see what he looked like. What he ended up doing was he came up with this box. And a lot of us use, it's basically the Punnett square. So when we do that great monohybrid cross, we line up the different alleles, And then we combine them to see what is the probability of the various progeny that you can get. Now, as a good thing to also note is that this is the probability with each individual offspring. So it does change, like not that it changes, but it does, it is the same probability for each offspring. So if you have 3 children, you are not guaranteed that you're going to get all dominant children each time, or if you get one dominant, that all the other children are going to be the either heterozygous or recessive. But you're going to get, maybe for one child, you might get dominant, you might get dominant again, etc., depending on the children. So again, looking at Mendel's plants, if we want to do the Punnett square for the plants, just a couple things to point out. We want to use the monohybrid, so that would be this one. You don't have to use it, but it just makes it more interesting for the Punnett square. And again, we can use big T, little t. I also make my lowercase symbol a little bit fancier or more cursive-like. The reason why I do that is because some letters, such as, I'm trying to think of a good one, S, it's really hard to tell if you don't write it correctly which one's big, which one's lower. So I usually do that so I really can distinguish it and so I don't make a mistake, especially if I have a timed test. So there we go. This is my results that I would get. And hopefully you can see that I would get 3/4 phenotype of dominant, which is what he got, and 1/4 phenotype of recessive, which is what he got. Now that we covered the law of dominance, the other thing that was really interesting is the law of segregation. Basically, that this is stating that different alleles will separate or segregate during gamete formation. Again, Mendel did not know about genes. He did not know about chromosomes. He did not know about how things separate, or meiosis, how things separated when you form the gametes. We now know that, and of course it kind of makes sense. But at his time, he didn't understand that or know that part. But what he did know is that the traits did separate in gamete formation, and it was kind of random how that happened. So here we have an example that, again, there's purple and white flowers. We have the dominant traits here. We get the hybrid. And basically, when you're making your Punnett square, you're kind of showing these are all the different combinations you can get. This is what you can get in each sperm. This is what you can get in each egg. And depending on which sperm meets up with which egg is really random. It's part of that law of segregation. The other law that he came up with was the law of independent assortment. So basically, this is saying that if you are looking at multiple traits, so things on different chromosomes, again, he didn't know about chromosomes, but we know about them today. He said that each trait will get separated into 2 gametes, but how they split up really has no bearing on the other one. So let's look at meiosis, all right? So here we have meiosis. We have 4 chromosomes here, 2 pairs of homologous chromosomes. And if they line up during metaphase slightly different, they're going to separate into the gametes different, dependent on how they line up during metaphase. And again, it's random how they line up in metaphase. In meiosis, the homologous chromosomes do find each other, but they can find each other and line up. So let's say in this case, the blue represents everything that contributed from the sperm. Here's everything that contributed from the ovaries. All the sperm contributions are on one side, all the ovaries on the other side. When it separates in meiosis, it separates here, you end up getting these combinations that you see for the gametes. And I'll just write that out just to show you. We have big R, little y, big R, little y, little r, big Y, little r, big Y. And then if, on the other hand, you lined it up slightly differently, so it's mixed, how it lines up in metaphase. Again, this happens randomly. You end up getting this combination and this combination in meiosis, so you end up in the gametes, the combination of little r, big Y, little r, big Y, big R, big Y, and big R, big Y. Now, why did I sit there and point out how the different combinations ended up? hopefully, or maybe, you'll understand and see that these are the different combinations that you can get when we do a dihybrid cross. Okay, so dihybrid, we're dealing with two, and we're dealing with differences of the alleles for each one. So kind of going back to Mr. Reginald Punnett, we're dealing with a dihybrid cross with these two genes, and we're going to have the different variations of each gene. We're not going to use this type of box for it. Instead, we're going to use this type of box. And one of the things that usually comes up when we have a dihybrid cross, as everyone always asks, or a lot of people have issues, is how do you separate out the alleles? And what I just kind of want to go over is the way I like to do it is I like to kind of think of my FOIL method. All right, so what I'm going to do is I'm going to do good old-fashioned FOIL. I'm going to get the first most different genes, so big R and big Y. I'm going to get the outermost ones, so it should be big R, little y. I'm going to get the innermost, so little r, big Y. And then I'm going to get the lasts. There. And I want to point out that those combinations I got, big R, big Y, little r, little big Y, little r, big Y, and big R, little y, the reason why I've got those, and actually I think I screwed up on here, that should be a lowercase. Let me fix that. Oops, need to be an eraser. So that should be lowercase. Those are the same alleles that we put on those dihybrid cross boxes. That's where those combinations come from. It comes from the possibility of how things get separated on the metaphase plate and get separated into the little gametes that you get over there. Okay, so we're going to do the same ones over on the other side. And let me get my colors back. Big R, big Y. Go yellow, big R, little y. My purple, lowercase r, big Y. This cool fuchsia color. And then you would use the square to help fill out the different combinations. When Mendel did it, he did it for different traits, and here is an example of him doing it with both the round seed coat, round shape for the seed coat of the peas versus the wrinkled, and the yellow versus the green. And again, he put up the different combinations that you can get. You had a 1/4 chance for each of those combinations. If you go back to that meiosis, you can see that those ratios, even though there were 2 of each one and there were 8 combinations, it would be a 1/4 chance. And if you fill it in, you would see that you would get basically a ratio of 9-3-3-1 phenotypic ratios. So you get 9 that were round yellow, 3 that were round green, 3 that were round wrinkled, and 1 that was wrinkled green. This 9-3-3-1 is actually kind of an important ratio to remember in dealing with a dihybrid cross. Because it tells you that things all kind of worked out okay. That is an example of a dihybrid cross. Lots of different reasons why that's important, but that's a kind of a key ratio you would get for this type of cross overall. Now, you do not have to always do a Punnett square when you're doing a dihybrid cross. In fact, it might be easier to use the rule of probability instead of doing that cross. I don't have a dihybrid cross here, but I have a tetrahybrid, not even a tetrahybrid, a 4-gene cross here. And I thought I can show you the rule of probability and how to do this to see if you can figure out what is your probability from this parent, these two parents, of getting this particular genotype for the child. All right, so can you figure it out? I'll give you guys some time. See if you can get it. So I know you all know exactly how to go about doing this. You probably made a huge square to do this and figure it out, right? Don't have to. We can use the rule of probability. And for the rule of probability, I am going to do a Punnett square, but I'm going to do a really, really simple Punnett square. So I'm going to do the simple Punnett square where I keep in mind a couple things. Law of independent assortment, where basically this gene combination is not going to influence this gene combination. So how do I do this? Well, let's say I take just the A's. What's the probability of having a child that's going to be big A, little a from those parents? Well, if I set up that little Punnett square, hopefully you'll see the probability is going to be 1/2. Because it'll be this one and this one, that's 1/2. All right? So now, what's the probability of getting big B, little b from this cross of big B, big B, big B, little b? Again, I'm going to start another little Punnett square because I find it easier that way. And sometimes you can do these Punnett squares right in your head, and that is totally okay. So I can see this is also 1/2. All right, let's do the next one. We have big C, little c, and then we have little c, little c. So big C, little c, big C, little c, little c, little c, big C, little c, and little c, little c. So I'm looking for big C, little c. I see, if I'm not mistaken, that is, nope, big C, little c, and big C, little c, also 1/2. I think I got that right. I'll double check that in a second. All right, and the last one is big D, let me do the correct letter. Big D and little d. And what's the probability of getting big D, little d? If I did this correct, it should be 1. All I have to do then, once I get all those, is just multiply that out. So yeah, if I multiply that out, it should be 1/8, or if you did your calculator, 0.125. All right, so that's just another way of doing it. If you're constrained for time, hopefully you can make a couple of these Punnett squares really, really easy, and you can get that done pretty easy and straightforward on that one. Okay, so here's another little story, a little situation. This is something that has happened. I have O negative blood. My husband has that's supposed to be positive blood. I made a mistake in my typo. Let me fix that right now. O positive blood. My children will definitely have O blood. I know that because O is the recessive trait. So if I look at the Punnett square for that, I and O, it's going to give me only O blood for my children. I'm not going to have you guys find out for you. But what about the Rh factor? So the Rh factor is a different gene. So first let's talk about that too. When you hear someone say O positive, O negative, or A positive, A negative, the two things you're talking about are two different traits, two different genes. It's like saying that someone has blue eyes and brown hair. You can have blue eyes and blonde hair, blue eyes and brown hair, blue eyes and black hair. You're dealing with two different traits, so it's two different genes. So the Rh factor, if you don't know, negative is recessive, but positive is not. So again, what will my children be? Well, we know that my husband has at least one positive dominant trait. We don't know what the other one is. So what we're doing is by having the recessive individual mate with the positive individual, you're doing what's called a test cross. We know that the recessive will always give the recessive allele to the dominant. If you end up getting all children that are dominant, and you usually would have to do a very large cross to do this. You need lots of kids to really check this out. You can't really just have one kid and be like, oh, yep, the kid came out positive, that therefore this must be Rh positive. If you have hundreds of kids and all 100 kids come out positive, then we know, oops, so if they all came up positive, sorry, like this, then you know that this would be Rh positive. If, on the other hand, you do this cross and you get one child, and again, if you do 100 crosses, you would probably get 50-50, but let's say you aren't doing 100 crosses. Let's say you just had one kid, and one kid came out, oops, sorry, that should be negative. I did that wrong. Should come out as a negative child, then you know that that second allele was a recessive trait and not the dominant trait. So a test cross is basically when you have an unknown, a heterozygote or a homozygous dominant, you don't know, and you're mating them with a known, which is a homozygous recessive, and looking at the offspring to see if the second allele from the original was either homozygous dominant or heterozygous. Now, some genes are linked. So we talked about the law of independent assortment, but some genes are on the same chromosome. So when I said the law of independent assortment, I was talking about on different chromosomes. One chromosome is not going to influence the other chromosome. But when we talk about linked genes, these are genes that are on the same exact chromosome. So they're always inherited or frequently inherited together. I shouldn't always say always. Let me just underline that or highlight that. It's not always. It can be, I'm going to change that to frequently inherited together. Okay, what I just took was there's actually a really cool database. You guys have access to it. It's free to the public. It's called OMIM, Online Mendelian Inheritance something or other. I forget what the second M is. But what it has every disease that has been identified to a gene and has them located. It has a lot of information about the diseases. And I just looked at one chromosome. So I looked at chromosome number 1. Now, this is just one part of chromosome number 1. And I was just looking at what genes were linked together with this particular chromosome. So here's, again, a small section of it. This is so much more here. And you can see that certain genes are on the same chromosome, so they most likely be inherited together. Things like myopia, which is a nearsightedness. Like this psoriasis susceptibility. It doesn't mean that if you're nearsighted, you are going to get susceptibility to psoriasis, but if you had a mutation in this gene and a mutation in that gene, those will get linked together and passed on to future generations if they're both mutated because they're on the same gene. They're also going to be fairly close to each other. This is inflammatory bowel disease, dyslexia susceptibility. I'm pointing out things like cataracts, also melanoma. These are things that most people are totally have functioning normal, and just because you have it, maybe you're functioning normally of this version of the gene doesn't mean there's not another gene someplace else that might affect it. But again, these are just some diseases that they have linked to certain genes. I liked, this one, cataract. Multiple types, and then it's linked to the Rh factor in Scottish families. These are all based on the research that they have, but it's kind of cool to see which genes are on which chromosomes, where they're located. And again, if they're on the same chromosome, they will most likely, if you have a mutation to both of those genes, they're most likely going to be inherited together. Now, I changed the wording on the last one. I said that linked genes are always inherited together, but then I changed it and said no, frequently. The reason why I did that was because something occurs during meiosis, and it has to occur during meiosis. If it does not occur, really you can't continue with meiosis overall, and that's called recombination. So remember our homologous chromosomes. So here's our parents, right? And we have two genes we're looking at, big A, little a, right? And you get your gametes form here, and we get our lovely heterozygote in the F1 generation. What's going to happen is crossing over always happens in meiosis. Now, crossing over will occur here, but if you cross over here, and what ends up happening is you recombine, so you would get a new gene that has big A, and you cross over, well, it still has big B. It's the same genotype, so you don't usually notice it. But if you do a crossing over event here, so we can get, during meiosis, you should get during gamete formation, you should get a chromosome that says big A, big B. You should get one that says big A, little b. And you should see here that that is what we get. Big A, big B, little a, little b. That's normal. But if crossing over occurs, then what's going to happen is you're going to end up getting a chromosome that's big A, little b. Big little a, big B. And that's shown here. Big A, little b, little a, big B. We call those recombinants. So one of the things I want to kind of impress on you guys and get you guys to understand is that your DNA and your chromosomes aren't non-changing. There's always exchange of DNA occurring in your chromosomes. Your DNA, as amazing and wonderful as it is, through meiosis can have alterations occur to it. And this alterations add to a lot of the diversity we see in life. It's actually one of the whole points of having sex is to increase genetic diversity. And the increase in genetic diversity is through that process in meiosis that results in crossing over. That's one way. There's actually other ways that our DNA changes, but I want to just talk about this one right now. So what happened, and the reason why they kind of figured this out, was when they looked at some crosses. At first, you get these crosses, and you would get lots of that were what we expected from this dihybrid cross. So in this particular cross right here, you expected to get, again, big A, big B with little a, little b. That's what you got. Little a, little b, that's what you got. Fantastic. Everyone was happy. But then there was always a few that came out weird. We call those the recombinants. And there, if you notice, it was a really small amount. It was less than a quarter. And again, probably initially everyone's like, oh, it was probably a mistake or whatever, and kind of pushed it aside. But then they started really looking at it, and they realized that this was occurring because of that recombination effect that was occurring here that occurs rarely. So again, during meiosis, you have this occurring during prophase 1. Your homologous chromosomes have to find each other. They have to, they usually have a crossover event occurs. It's rare, but when it does occur, it does add some genetic diversity to our progeny. Now, we can use this recombination and this numbers that we get to help find out how far away genes are in the genome. This is one of the most older methods that we had of trying to figure out where genes were on the genome. Now we can just take DNA, sequence it, and it's actually really, really cheap to do that. But this is historically something that they did in the past, and I just want to kind of give you guys aware of it in case they give you one of these types of questions. Again, on the AP exam, they have done it before, and I just want to make sure you guys know about it, and just in case. So basically, the idea of it is that the further apart the genes are on the chromosome, the more probability there is of crossing over to occur between these two genes. And so we can look at the recombinant phenotypes and calculate the crossing over events between those, and then figure out how far apart these genes are on the chromosome. Does that make sense? So basically, the number of recombinants is directly related to the distance it is on the chromosomes. And a map unit, so one map unit is going to equal the crossing over events, or the percentages, basically. And one map unit can also be represented as one centimorgan, named after Thomas Hunt Morgan, who first did a lot of the work with fruit flies. So kind of like looking at this one, you can see that the parental phenotypes, and usually the parental phenotype is the one that you see the most of. We have 944 and 965. If I was to do some quick calculations, I would tell you that the percentage of the parental phenotypes would be, I could do that right now, 944 plus 965 is 1,909. And then of my recombinant, I would see that there was a total of 391 recombinants. I'm going to add these two together to find out how many total we were working with. So the total amount of fruit flies that were looked at is 2,300. Again, that's such a beautiful thing about fruit flies, is you can get so many progeny to look at in such a short period of time, which is great when you're doing genetics. And then when I look at the percentage of recombinants, I can use that to figure out approximately how far away the black gene is from the vestigial gene. So I would take 391 divided by 2,300 to give me percentage. Oops. And that would give me approximately 0.17. That would be in percentages 17%. And so I would say that these two genes were 17 map units or 17 centimorgans away from each other. Okay? So here we have a problem. It is a crossover frequency of each type of gene on a chart, and they want you to find a chromosomal map for these particular genes. So again, we'll just kind of clarify, we're dealing with 4 genes, A, B, C, and D, and using the frequency of crossover, try to figure out relative distance that they are on the map of the chromosome. So let's say this is my chromosome. How far apart would they be from each other? So how do I go about doing this? Well, the first thing I would do is I take the two genes that are the furthest away from each other. So looking at this one and looking at my crossing over events, 45%, so these two are 45 centimorgans or 45 map units away from each other. Remember, centimorgans and map units are the same exact measurement. So I would put those on my fake chromosome here that I drew. I would draw them as far away from each other, and then I would kind of maybe put a little marking for me to know that they are 45 away from each other, 45 map units. Then I got to figure out a couple other things and do a little bit of figuring things out. So if B and C are 45 map units away, B and D are 40 map units away. Now they can be in two different places. My D can be over here 40, it's quite possible, or it can be over here 40. Okay, so I'm going to put my B and D here for now. Put D over there and write down 40 for that, okay? Knowing that it could also be over here 40, so we'll kind of put that in question mark, okay? We're going to see if everything else kind of fits. A and C are 30 map units away. All right, so if A and C are 30 map units away, again, A can be over here for 30, or it can be all the way on this side for 30. We're not really sure. Okay, so we'll use some other data to help fill this out. So we got A and C, we got B and C, we know that. A and D are 20 map units away. So let's do a little math here. A and D, so we know that. So if D was over here, it would be 40 away from B, and if A and C is 30, we would see that A and D would not be 20, right? It would probably be, because B and C is 45, and A is, even if A was over here, we have this would be too big of a gap. So that's going to get rid of this for me. I know that at this point, that gives me the information that D has to be in between B and C. Okay, so I'm going to check that off as something that I know right now. All right, now, that being the case, A can now be either on this side or this side. So let's say A and C is the 30 over here. If that was 30 and then we have 5 here, that would be 35 distance from A and D. And is that what we got for distance? Nope. So that also tells us that we can't have it in this location. So that kind of gives us the idea that A and D has to be located over there someplace. And that kind of gives us the relative distance of everything from each other. So we have B, A, D, and C is the distance. And again, if we want to kind of fill things out a little bit, we know B and C is 45. We know that this is going to be 5 because we have this is 40, so the difference here should be 5. And you might see that you get a cross that would say D and C was 5, and that would work out great. At that point, we'd also have to figure out what D and A is. So now we have A and D is going to be 20. That's already given to us. Now you might notice that A and D together is going to get 25, and A through C should be 30. It's a little bit off. Be aware that sometimes these crossover events are not perfect numbers. That's actually a pretty big number that's different that I'm not too pleased with. Usually I would get like maybe 19 versus 20, and might be differences in math. But that would be probably the best we can get at this point. So do be aware that does happen sometimes, and the reason why that happens is because the way they do these crossover events is they actually do fly matings. And so that kind of throws off our numbers a little bit, but I think that would be an okay answer for this. But the idea of it is you just basically kind of want to piece it together as best as you can, knowing that it can be either side. I also want to point out something else. This answer, B, A, D, and C, is the same as C, D, A, and B for G order. Just a reminder that chromosomes do not necessarily go from one side to the other. They move around in circles. So C, D, A, B on a chromosome is the same thing as if that chromosome flipped over and was B, A, D, C. I have seen this type of question on an AP exam much more frequently than something like this type of question. So they're mostly going to do something to this effect if they ask you. But again, I want you to just be prepared of what it is, just in case. So now let's talk a little bit about non-Mendelian stuff, and we're going to talk about sex-linked traits. So this is non-Mendelian. All right, so you probably are aware that you have autosomes and sex chromosomes. And basically, sex chromosomes are chromosomes that determine the sex of the individual. Now, as you can see here are two X chromosomes. They're big chromosomes, and you have the X and the Y. This is a really short chromosome. So what ends up happening is if you are a male and you have a recessive gene somewhere in the X chromosome that does not have a partner on the Y, then you only need one copy of that gene for it to be expressed. So that's one of the things that we see is that traits that are carried on the sex chromosome that might be expressed because there's only one copy of it in the male tend to be linked to the sex, in this case would be male. Be aware there are other ways that other organisms have different sex chromosomes. It's not X and Y. If you do get a Gen X question like this, they would probably explain how the X and Y works or the sex chromosome works in those organisms and ask you to kind of extrapolate what you know about this onto the different organisms. There's some great chicken sex-linked chromosome problems out there. If you want to search that to kind of get practice on it, it's a great resource to make sure you understand how to extrapolate into a different organism. But again, this was all found in fruit flies originally, and now we kind of extrapolate it to other organisms. But what we've seen is that here's an example of a sex-linked trait on a fruit fly. We have white-eyed, and so the white-eyed is expressed as a dominant here with a big W versus the lowercase for the recessive. You had a white-eyed female mated with a red-eyed male. And the big thing to do is when you do these crosses, make sure you put the X and Y. I would not write these as like a box like this, a Punnett square like this, because you're not going to know what the sex is and you're not going to see the sex linkage. So that is something I would not recommend at all. Instead, I would look at making sure you have the X and mark it with a Y, or W, to represent the allele in this case. Separate out, you have the females on one side, you have the males on the other side, and when you mated them together, you would see that you would get red-eyed females and white-eyed males. And again, when you see all of the males have a trait, then you can kind of look at it and say, uh-oh, this might be a sex-linked trait. And that's what you want to look for in these types of problems. Now, because of the two chromosomes that females have, what ends up happening is one of them gets inactivated, and it's a random event of which one gets inactivated in the females. You can look at some of the cells and actually see it. So here's a nucleus right here. This right here is the nucleolus. And you can see this little black thing right up against the membrane of the nucleus, the nuclear membrane. That's a bar body. If you notice, these are males. They don't have it. They have the nucleolus there, but no little condensation by the membrane. And again, this is explained really well and nicely in calico cats. All calico cats, except for a few exceptions, are females. And the reason is to be a calico. It's not a breed, but what it is that there is the gene. This is a sex-linked gene. It's associated with fur color. So here you have the orange allele, you have the black allele, and during cell division, randomly one gets inactivated. So the black fur here has the X is inactivated as the orange one, and the brown or orange fur here has the black fur gene inactivated, and it's random. You can see that all the cells from that inactivation ends up being that color. So that's what you see when you see that tortoiseshell or calico color. It's because of X inactivation. Another inheritance pattern that is not following other inheritance patterns that don't follow in Mendelian genetics is things like incomplete dominance. At the time when Mendel came up with this theory of genetics, his idea of genetics, a lot of people thought blending occurred, meaning if you had a red flower with a white flower, you'd get a pink flower as the progeny, the offspring. And that does happen sometimes, and that's a form of incomplete dominance. What we now know, now that we know genes and we know about DNA, is that in proteins, what we know here is that this flower is making the red pigment, and it makes a lot of it because it has 2 copies of the red pigment. This flower is not making any red pigment at all. When you mix them together, now you have half of the amount of red pigment being made, and that's why you end up getting that pink color. So that's why you have that incomplete dominance. Codominance, on the other hand, is different than incomplete dominance. Incomplete dominance, you're going to see a blended pattern. Codominance, what we're going to see instead, is we're going to see not blended, but you're going to see each individual parental you're going to see the parental trait individually in the progeny. So here you have a white flower with a red flower, and you don't get a pink, but you get white spots and red spots. That's what we end up seeing with codominance. Another great example of codominance is the ABO blood type. A and B are codominant. So that means that you don't see A, you don't see B only when you have both alleles. You see AB proteins on that. And another thing that we can see is something called polygenic inheritance. Again, poly means many, genic means genes. There are some traits that are influenced by many genes, and they don't give you a distinct red or white. Here's an example of skin color. We have lots of genes that are associated with that. Here we're showing it simplified as just being 3 genes and the different alleles. And if you have a heterozygous of those 3 genes and different alleles, you can get lots of different combinations, and that gives us a really beautiful bell-shaped curve in terms of what you can get in the population. So when you see some things like height is a great example of that, you don't see a distinct yes or no, that is a good example of probably a polygenic inheritance is involved. Lastly, there's non-nuclear inheritance. Hopefully you guys remember about the endosymbiotic theory, how the mitochondria was at one point a bacterial organism that got picked up by another one cellular organism, and they had a symbiotic relationship, and now we all have mitochondria that have its own DNA and its own ribosomes. But mitochondria is also passed on through the mother only. So because of that, if there is a disorder associated with the mitochondria, the DNA in the mitochondria, you're not going to see the normal Mendelian genetics pattern. You're going to see that gets passed on to the child no matter what the father or the paternal mitochondria is or genes are, because of the fact that only the mother's mitochondria gets passed on to their children. You may need to look at pedigrees, and I just want to kind of show you what a pedigree is. Because humans, again, we don't like to purposely mate humans to see what their children are. People get mad at us for that, and geneticists won't force people to mate to look to see if they have a genetic disorder. But they can look back at the history of the family to see if there's a genetic disorder, what the trait is, the inheritance pattern of this particular genetic disorder is, and etc. So this is a famous family. It's the royal family. You may have heard of William and Harry, Prince William, Prince Harry. This is Kate. Two of their children are here. There's actually a third child. We all know about Louis. And then also Harry's now married and has a kid too. But there was at one time a disease that was ravaging through the royal families, and that was hemophilia. And again, you can see those afflicted shown here. But basically the overall is when you're reading a pedigree, each level is represented by usually a Roman numeral, and the first level is 1, second level is 2, we have 3, 4, 5, 6. So Philip and Elizabeth would be 7, and Charles would be 8 in this pedigree. Kate and William would be 9, and finally their children is 10. So we have 10 generations represented in this particular pedigree. And if you were asking questions, you would always identify, which generation you're dealing with and which person. So again, starting from left, it would be 1 and 2. This would be 1 and 2, exactly, just like that. Shown like this. So you kind of know who you're talking about for each generation. A female is shown with a circle. A male is shown with a square. And if you are afflicted, you are colored in. If you are maybe a carrier, you might be halfway colored in. So in here, we don't have any male carriers because this is a sex-linked trait with hemophilia. So let's just get a little bit of information of how that is represented overall. Now, besides being sex-linked, another thing that is not influenced by Mendelian genetics, something that doesn't follow Mendelian genetics, is environment and traits. There are some traits that are some of our genes are influenced by the environment. Things like this phenotypic plasticity. These are lovely hydrangeas. They are known to have different flowers based on pH of the soil. And sometimes you can have the same flower in the same exact area, but the roots are in different parts of the soil that has different pH, and they'll come out in different colors based on the pH of the soil. Some can go from white to pink to purple, all based on that pH. Here we show pictures of 2 rabbits. Those are Arctic hares. They actually have a change in their fur color based on the season. And again, that's affected by protein expression, and the protein expression is based on light coming in. So as their photo period, how much light they get during the day influence whether the pigment for the brown color will get expressed and made, or if they're getting less light during the day, that sends a signal that they're not going to make that brown pigment. You may be familiar with things like height and weight can definitely be influenced by environmental factors. If you don't have enough nutrition, you're not going to get as tall as you could be. If you've ever gone and seen the old houses from New England times, and the beds are tiny, and part of the reason is because they didn't get as tall in the 1800s because they didn't have enough nutrition, and sometimes having too much nutrition can affect your weight. And then also what's interesting is sex determination in reptiles is influenced by temperature. For example, on alligators, if there is too high of a temperature or too low of a temperature, they're going to produce more females. And if it's just the right temperature for them, they will produce more males. This is actually a big deal because as the environment is getting warmer, we're seeing that the ratios of these animals are producing more males and not as many females, which overall affects also the number of progeny they're going to make. Now, we've talked a little bit about meiosis as I've gone through some of the genetics. I just want to highlight some of the key points because, again, I'm assuming most of you guys have got meiosis before, but the big thing to remember is that meiosis allows crossing over to occur. That occurs when in prophase 1, where you have homologous chromosomes find each other, they line up, and they're going to exchange genetic information. Again, kind of showing here, this eventually ends up being a paternal trait. This ends up being that recombinance, and those are usually rare, but we do see those in the progeny. The other big thing to also remind yourself about meiosis and how it adds to genetic diversity is that independent assortment. Depending on how the chromosomes line up in metaphase really depends on what ends up going into the gametes. And there's all these different combinations that can kind of occur. So because of these two things, it really just shows how unique you really are when it comes to all the different things that could be possible from meiosis. And the last thing that can also add to genetic diversity is non-disjunction. Here, basically, what happens is in meiosis, at one point, maybe the chromosomes don't separate out correctly. So here we have two homologous chromosomes, and during meiosis 1, they both end up in the same cell. This would end up being a gamete that would be non-viable. And then these get separated out, and you can see that there's an extra chromosome here that's not supposed to be here, and that ends up being a trisomy of that particular chromosome. Another form of non-disjunction that can occur is if it occurs during meiosis 2. So the homologous chromosomes separated out perfectly fine, and these end up continuing totally okay. But then during meiosis 2, you end up getting two of the sister chromatids on the same side, and again, that can give you a form of trisomy. So here's where you might see some complications that occur, and again, this would be a non-viable gamete. And finally, we can also get things that occur in meiosis that can add to chromosomal abnormalities. I told you that crossing over occurs in meiosis, but sometimes the crossing over can occur not in the best way. So here you can see that it's not there's a little loop that was kind of formed in that process. If you have this loop form, and again, it's random how it happens, once that crossing over occurs, if I was to follow this, right, so here you have A, B, and you would have the crossing over here, and it wouldn't go necessarily CDA, well, you could get the CDA, but you'd actually probably go A, B, and you might do B, E, F, G, H, and that's what you see right here. And that could be viable because you have all of the genes in there. You have another one that's totally viable. That's going to be over here, okay? But you're going to get some that are not viable because the combination just came out. Either you deleted a huge chunk of the chromosomes or you duplicated a huge chunk of the chromosome. And that would lead to a non-viable gamete at that point. So those are things that you have to worry about during meiosis and genetic differences. Hopefully you got all that in. If you have any questions, feel free to reach out, and see you guys soon.